Welcome to the Topics in Rehab Science Lecture Series, the Shrier Family Topics in Rehab Science Lecture Series. I'm delighted today to present Dr. Amy Lebkuecher. Amy is a postdoc in my lab and in the lab of Branch Coslet at Penn, and she also now works in Erica Middleton's lab as a research associate. So she's a very busy person. And Amy did her doctoral training at Penn State, where she got a PhD in psychology and language science. And she also has some clinical background. She worked at the Kessler Foundation for a while. Her thesis work was in the interface between language and non-linguistic domains, particularly motor planning. And her work that she's continuing with us is continuing to be at the interface between language and action. So welcome, Amy. Thank you. I forgot to say, sorry about that. If you have quick clarification questions, and you guys indicated that you should feel free to interrupt. Longer discussion items can be saved to the end. Thanks.
Thanks for the introduction, Laurel, and thanks to everybody for coming out on this truly frigid January day to my talk. I appreciate it. So today I'm going to be talking about a project that we have going on looking at action semantics in primary progressive aphasia and now also Parkinson's disease. Figure out how to press the slides. Not sure what's on it, but today's Shrier Family Topics in Rehab Science lecture is supported by a generous gift from Nancy and Marc Shrier, who we're grateful for their support and belief in our mission. For those of you who are doing this for continuing medical education credit, CME, this is the code, and I will post it again at the end during the discussion section, so it will be visible for anyone who needs that information. I don't have any financial relationships to disclose. So the learning objectives for today's lecture, by the end, you should hopefully be able to identify key differences in semantic impairment between primary progressive aphasia and Parkinson's disease, as per the current literature on these subjects. You should also be able to describe what each you may be able to add to our understanding of action semantics across these populations. And the third one is a little bit tentative because our current results are quite preliminary, but hopefully you'll be able to describe what the study findings suggest so far about the potential utility of hand gestures as a tool for language rehabilitation across these populations. So to understand language, we rely on more than just speech. We also glean information from hand gestures, from facial expressions. For today's talk, I'm going to focus on the integration of hand gestures and speech in language comprehension. To understand the intersection between language and gesture, we need to consider the brain. So, as many of you know, language and action rely on a broadly overlapping network of regions in the left hemisphere, consisting of a dorsal route, which maps more directly between input and output, and also a ventral route that taps into semantic memory, which is our representation of meaning and long-term knowledge about the world around us. So to give you an idea of what I mean by semantics, let's consider how your mind represents the concept of a hammer. So some of the concept consists of what a hammer looks like, for example. You also have a concept for what a hammer feels like and how you need to shape your hand and move your arm in order to successfully use a hammer. There's also semantic information which isn't a direct property of the hammer itself that you learn based on co-occurrence, such as objects that are often associated with hammers like nails, and contexts where hammers are often used, such as the workshop shown here. But of particular relevance to the connection between hand gestures and language and today's talk and the current project, action semantics are of particular interest, that knowledge about how to use them. So for the purposes of the current project, we define action semantics as semantic information about actions that is accessible to language and during execution, observation, and mental simulation of action. An aspect of action semantics that is currently up for debate is whether the activation of the sensory motor system is important for action semantics. To investigate this, we are examining action semantics in clinical populations, both with and without a disruption of the sensory motor system. Specifically, we are studying individuals with Parkinson's disease, a neurodegenerative disease with prominent motor deficits, and primary progressive aphasia, a neurodegenerative disease without prominent motor deficits. Not sure why the title isn't showing up for that slide, but it's supposed to say study aims. So the current aims for the study are, and these are broad aims for the study as a whole. So we're not necessarily going to address all of these today with our preliminary findings. But we want to evaluate action semantics in individuals with PPA and Parkinson's by investigating whether meaningful gestures facilitate language comprehension in these populations. And then within that, we also want to examine whether the activation of the sensory motor system plays a key role in action semantics in individuals with PPA. And then additionally, we'd like to understand the characteristics of individuals in these populations who might benefit from observing meaningful gestures during speech language therapy. So here's a roadmap for the rest of today's talk. I'll begin by reviewing the existing literature on action semantics in PPA and Parkinson's disease. I'll also describe the relevant EEG components for examining action semantics in the context of this study. We'll talk about the current study's hypotheses and methods, and then we'll discuss some very preliminary findings and what conclusions we can potentially draw so far and future directions that we're going to continue to explore as the study continues. So for those of you who aren't familiar, PPA, primary progressive aphasia, is a neurodegenerative disease that's characterized by substantial language function that occurs early in disease progression. And some individuals with this disease also exhibit impaired semantic abilities, so impaired long-term knowledge. There's some evidence that individuals with PPA have limopraxia, a condition which results in difficulty performing skilled actions such as hand gestures. And additionally, limopraxia in this population appears to be characterized by deficits in action knowledge for at least some individuals with PPA. But overall, there's not a lot of research that looks at action semantics in this population. Parkinson's disease is a neurodegenerative disease. This one affects the frontal striatal circuit and is characterized by prominent motor deficits. Individuals in this population end up with indirect effects for frontal and motor and sensory motor areas as a result of their connections to the basal ganglia and the dopaminergic pathology that's characteristic of the disease. So in terms of the action semantics literature on Parkinson's, there is evidence that individuals with Parkinson's exhibit comprehension deficits for action-related words, even early in disease progression, and notably also in the absence of other cognitive impairment. This is considered by some to be evidence for deficits in action semantics, but this interpretation of the deficits are not necessarily agreed on by all. So overall, the current state of the literature suggests that there's a need for more investigation of action semantics in these two populations. So EEG is a method that's been used widely to investigate language and semantics and cognition more generally. It is a very useful tool given that it has higher temporal resolution than other tools, so you can see activity from the brain locked to a specific stimulus in a specific moment. And it does not inherently require participants to produce an overt behavioral response to see differences in responses to stimuli, for example. And you can imagine that when you're working with a population that struggles with language, this could be a potential use. So one EEG component of interest, there should be a title that says N400 there, but once again, I'm not sure why it doesn't show in presentation mode. But this component of interest is called the N400. It's a very commonly studied EEG component. It's characterized by a greater negativity in the EEG signal at approximately 400 milliseconds following an unrelated or unexpected stimulus. So for example, in the current figure, the greater negativity of the unrelated words versus the related words here. And this component is of interest for the current study because it is considered to be an index of semantic processing. So in addition to the N400, we are also interested in the desynchronization of beta and mu oscillations. So these particular oscillations are of interest because they are generated by brain regions in the sensory motor system. So a decrease in the amplitude, or another way of putting it is desynchronization of these oscillations, suggests the activation of regions in the sensory motor system. So we can examine this desynchronization to examine the involvement of the sensory motor system in action semantics. So based on the findings from previous research, these are the hypotheses for the current study. So we hypothesize that at least some individuals with PPA and Parkinson's will exhibit a reduced benefit for language comprehension when they observe meaningful related gestures. And this would stand in contrast to prior research with healthy individuals, where they show a benefit from observing these meaningful gestures. We also hypothesize that these particular individuals, which would exhibit broad semantic impairment and also potentially deficits in gesture recognition. We would argue that any reduced facilitation that we see behaviorally of comprehension should also be illustrated by a reduced N400 effect. If sensory motor activation is key for action semantics, then disruptions to the sensory motor system, as evidenced by reduced desynchronization of beta and mu, will be related to reduced N400 facilitation effects. So the findings that I'm presenting today are extremely preliminary, and we won't yet be able to draw firm conclusions regarding these hypotheses, but I just wanted to give you an idea of the specific goals of the overall goals of the project. So the overall recruitment goal of the current study is to analyze data collected from 10 individuals with BPA, 10 individuals with Parkinson's, and 20 age-matched neurotypical controls. So these are small sample sizes. One, because these populations can be a little bit difficult to recruit, and primarily because we are exploring some pretty novel territory with the current study. And so we want to do a small pilot study to examine the feasibility of our approach and to hopefully get some preliminary data for a larger study in the future. Today, of that overall goal, we currently have data from 3 individuals with primary progressive aphasia, 1 individual with Parkinson's. We started that component of the project a little bit later, and then also 5 age-matched neurotypical control. This is an illustration of the EEG electrode array for any individuals who are interested in those details, and then is also just some of the methodological details of the software that I'm using for various components of the project. Happy to chat more about those details later with anyone who has suggestions or is interested. So for our main paradigm of interest, during the EEG recording, we have participants observe meaningful gestures, and then they hear a word. It would help if I actually used my stimuli that I put in the presentation. So they see a meaningful gesture, and then they hear a word that is either related, unrelated, to the previously presented gesture, after a brief pause, of course. So if participants are sensitive to the relatedness between word and gesture, then they should exhibit the N400 effect that I've been talking about. So in other words, a greater negativity for unrelated compared to related trials, right? Did you make me? We additionally have participants complete a behavioral version of the task where they are asked to indicate via key press as quickly and accurately as possible whether the word is related to the previously presented gesture. So you might notice that we are doing the behavioral version of the task separate from EEG recording. This is because of our interest in the beta and mu oscillations, because they often index preparation of the motor response. We didn't want any preparation of response during the task to interfere with any observed desynchronization that happened as a result of the stimuli being presented. So that is why we decided to separate out those components of good design.
Two sessions. This is in order to maximize the number of trials that can be included in the analyses. This EEG can be a noisy, and this is particular. In the third session, we have participants complete the behavioral version of the Narrative, as well as a battery of neuropsychological assessments that design assess language ability, semantic ability, and today's preliminary findings, we're going to focus on the session 1 EEG findings and also their performance on the battery of tests. Okay, so let's talk about some preliminary findings. So before I dive into the preliminary findings of the study, I want to touch on a slight issue that we saw with the accuracy in the main behavioral test with our neurotypical participants. So when we first visualized the data, as you see here, we were concerned that the accuracy, it was pretty high, but not exactly as high as we were expecting, particularly for the related trials. So on the x-axis here, we have participant mapped out. The neurotypicals are grouped together in this column NT, and we have the percentage correct on the y-axis, with related trials represented in blue and unrelated represented in red, which will be true for all of the visualizations that I present to you related to this. The asterisks just denote that performance in that particular category was significantly above 50%. So, they're doing reasonably well, but not quite as well as we'd expected, particularly for the related trials, which we expected to be easy. So thankfully, we had normed our stimuli before we conducted the main experiment with a separate sample of neurotypical. We asked them to rate how well each gesture matched the word on a scale from 1 to 7, with 7 being matches really well, and so for the rest of the data visualizations, we're going to focus on the target words where the average rating for the related gesture in the norming study was at least 4 languages than not. And here's the cluster of electrodes that we selected to examine the N400 effect. This was based on what we previously observed in literature, looking at the N400 more generally and also in action semantics. This is a table illustrating some of the demographic information from our participants, as well as their scores from the assessment battery from session 3. And we included the neurotypical group altogether in the first row for comparison to our individual patients. So we'll start with the ERP and behavioral results for the neurotypicals for the main task of interest. So on the left, we have the ERP results. Amplitude of the signal in microvolts is plotted on the y-axis, and you'll note that it's positive upward. The x-axis is time denoted in milliseconds. I know that the text is a little tiny and hard to see. This middle point is about 400 milliseconds. The average where we tend to see the N400 effect. We examined the two numbers on the graph represent the difference in mean amplitude between unrelated and related trials over 2 time points. So the first time window is a classic N400 time window of 300 to 500 milliseconds. We additionally looked at the later time window because older adults have been shown to demonstrate delayed N400 effects in prior literature. We wanted to make sure that we weren't missing any effects that happen a little bit later on. And so as you can see, there is some evidence for a little bit more negativity in the signal for those red unrelated trials compared to related trials. And additionally, neurotypical individuals exhibit high accuracy, greater than 90% across both related and unrelated trials on the behavioral priming task. They do exhibit slightly higher accuracy. The ERP graph here just for a visual comparison. The second graph here illustrates the response time findings from the behavior. So you'll note that unlike the other graphs, I plotted the neurotypical individuals separately here. This was because the error bars would be really difficult to interpret. write them together. But as you can see, the neurotypicals don't show a huge difference overall between unrelated and related trials in the response time. Some participants show a bit of a trend towards faster response times, but the differences aren't very. So now we'll turn to discuss the findings for our first PPA participant. As you can see from their assessment, they are relatively impaired in terms of their language, semantics, and gesture. So they show relatively broad impairment of the specific abilities that we're interested in. And you'll also note that their ERP signal is messier than that of the neurotypical individuals, and that it is a little bit difficult to pull out a consistent pattern here. So, whereas before, prior to 400 milliseconds, there was a pretty clear separation between the related and unrelated trials for neurotypical participants. Oops. That one. Here you can see that there's kind of a switching back and forth between the negativity. So not sure that we can draw too many conclusions from their ERP signal here. And additionally, you'll see that their accuracy here on the behavioral task is pretty low. Running this participant, they had extreme difficulty keeping up with the pace of the task since the behavioral task is a timed task. And they would, get hung up on the previous trial if they felt like they didn't properly perseverate the unable to. So unfortunately, that resulted in very low accuracy on their part. And as a result, I'm also not giving them the response time data because there just isn't a sufficient sample to get a clear pattern from the few trials. Yes.
Is it actually accuracy or just chance that it looks around 50% per hour? Yeah.
So, well,
it's just
yeah, I guess that's fair. So they're unrelated trials. To be fair, I should have examined whether this was significantly religious for the related trials, but it does appear that their unrelated trial accuracy is kind of restricted. Which, yeah, that's what you would expect, right? They are just kind of hitting buttons and they're not sure. Yeah. So our second participant with PPA is much less impaired across the board than our first participant, but does demonstrate some deficits in gesture recognition. So in terms of their ERP, we do see some greater negativity for unrelated compared to related trials. Obviously, not a whole lot of separation between the two trial types for this particular participant. But interestingly, we do see a very similar pattern in their accuracy on the behavioral task that looks almost identical to that of the neurotypical individuals. So it's interesting here that even though their ERP is seeming a little bit to diverge slightly less than that of the neurotypicals, we do see a very similar behavior. What's even more interesting, in my opinion, for this participant is that even though they show that high accuracy, their response time actually demonstrate a slight trend towards faster response time for related relative to unrelated, which is different than what we see in the emerging. So our third individual with PPA is not quite as severely aphasic as our first individual, but they also do demonstrate pretty broad impairment of semantics and gesture recognition if you look at their matter scores. Interestingly, though, when you look at their ERP signal, we do see a greater negativity from the unrelated trials compared to the related trials for this individual. However, their accuracy is much lower than that of the and so they're not performing particularly well on the behavioral task. And additionally, unlike the neurotypical individuals and the other two individuals with PPA, they're showing higher accuracy for related trials, and they're particularly doing poorly on the unrelated. And when we look at the response time is more variable than the other participants. And additionally, we see, like the previous PPA participant, the second individual, we see a slight trend towards a benefit more related compared to unrelated trials. Once again, these differences are not huge, the state is very preliminary, so I don't want to make too much of it, but it is interesting. So now we'll discuss the findings from our first participant in the study with Parkinson. As you can see from their assessment data, they do not demonstrate evidence for impaired language, semantics, or gesture recognition, despite that this participant has visible tremors. We asked this participant, and we will ask all of our individuals with Parkinson's disease to delay taking their morning dose of disease-modifying medication in order to test them in their off days and maximize the possibility of observing any interesting differences in our measurements. This is an approach that has been adopted in prior research quite a bit, and it does suggest that you're more likely to find differences. Yes?
Do you have any measure of their actual
I do not have that here, and I'm sorry. It is worth, let me write that down. I would say despite the visible tremors, they would drive themselves there, so that's an unofficial indication. But yeah, no, it would be good to characterize this person. So, as you can see, this individual, when you look at their accuracy, they, they actually slightly outperform the more typical participants in terms of their accuracy and behavioral tests. And their ERP findings do demonstrate a clear difference between related and unrelated trials. However, you'll notice that the polarity here is switched. So they're showing greater negativity for the related compared to the unrelated. So this could be an effect of disease, an effect of medication that they're on related to the disease. This is also apparent, it's very rare, but apparently some individuals will demonstrate a reverse polarity and positive effect. So that could just be the case for a particular person. So we need to collect more data before we interpret too much from it, but I am curious to collect more individuals with Parkinson's to see if this is a trend. Yeah.
I'm sure the procedure, the video, and then notebook planner.
Yeah.
Suggesting that the level of analysis is independent. But here, so it actually is the same item preceded by a related person.
Correct. Yeah, so the parents are right.
Okay, so the serving has its own.
Yes, I apologize. The way I framed that was a little bit confusing, but yes, each word is serving.
That's ideal.
Yeah. And then when we look at their response time data, I mean, one thing that's a little bit surprising is that overall they're not particularly slow, despite being tested off their medication. But additionally, they're not really showing much of a difference in response time. So looking a little bit similar. I mean, I see.
Oh, no.
Sorry.
No, I'm just thinking.
Okay, yep. I'm thinking about it too.
Like very
This person's very intelligent. Yeah, so they're looking, response time accuracy wise, looking pretty but yeah, their signal is looking pretty different. Yeah.
Do you think that they have primary details? They're all together?
I do not know that.
It'd be important too. This is where I was asking you about the motor, because your comparison is somebody who has versus one. PD has different phenotypes.
Right.
You want to make sure you're getting the phenotype that you have.
What did you say? Yes. There's no need for anything. Well, speaking of the history, with the, there are others lower than 3. So you would, I guess, the reason why I asked, you would probably expect more correction studies.
So, given how preliminary the findings for this study are, I'm really hesitant to draw any firm conclusions from the current data. But I am encouraged to note that the EEG signal across participants does seem to differ depending on whether the trials are related or unrelated. So that generally supports that our stimuli are, or at least to some extent, doing their job. And behavioral findings, and this is an extremely tentative suggestion, but behavioral findings from some of the individuals with BPA point towards a potential benefit of related gestures for brain function. So they showed a very, very slight trend towards shorter response time on the behavioral task for related neurotoxic trials. But once again, we need much more data. And then to draw firmer conclusions regarding the Parkinson's disease in this particular context, we definitely, need more than one participant, but additionally, it would be good to get some participants who exhibit comprehension impairments or semantic impairments. So a next step for the project is to analyze the desynchronization of the beta oscillation and the new rhythm. That way we can investigate the role of the sensory motor system and how it relates to our observed behavioral and ERP effects. So hopefully the next time that I present this project to you, in addition to having just more data overall, we'll be able to talk a little bit about data and news. So I just wanted to say thank you to my mentors and my speech language pathologist consultant, Pippa, my research assistants, and also Mitch Caesar for helping recruit for the Parkinson study, and Klein Family Parkinson's Rehabilitation Center of Excellence for funding the Parkinson's study. So thank you to everyone for your attention. And then here is the code again for those people needing it.
Most often easily or kind of healthy. Is that like a summary? But there was one person that
Yeah, so for the two of the individuals with PPA showed a little bit of an easier time on the trials that were related to each other rather than unrelated, at least in terms of response time. And additionally for accuracy related too. So the one person wasn't necessarily higher, but was pretty equal to the unrelated trials in accuracy. And the person with like pretty profound semantic impairments was surprisingly showed benefit for related trials. So this is something of interest. I've kind of started looking into the literature on like semantic priming, for example, because I think it's pretty relevant for the current study. Given that in semantic priming, we would also expect to see we would expect generally to see a benefit for related trials. But apparently there's also some evidence of a reverse effect if you have stimuli that are particularly different. So the suggestion, and I think this kind of fits with what might be happening here, is that for the unrelated trials, they are so unrelated. So that when they pop up, this is like the next step. And yeah, I think that's true too for our stimuli for sure. So I'll be interested to see if that continues, because it still is a little bit the case, even when we kind of narrow it down by our particularly, quality items in terms of the work.
So how is it that a person with language difficulties is
getting the benefits, finding it easier than a healthy.
Well, so I don't know that I would say that they're finding it easier, right? So their response times were longer and longer, very long, right? It's just the case, honestly, it could be the case that maybe the neurotypicals are not really struggling with the pairings that we've just focused, maybe because we looked at the high quality, these pairings in particular were a little bit easier for the neurotypical individuals, so they didn't necessarily show a huge difference by condition. But I think, yeah, for the individual with semantic deficit, they didn't have an easier time. Their accuracy was lower, the response time was longer. It could be a factor. There's also information in the literature looking at hyperpriming, which is a phenomenon that's sometimes seen in other clinical populations where you actually do see kind of exaggerated priming effects for individuals who have some level of semantic impairment. It's not something that I've looked into. I didn't put it in the discussion because it's something I've only pretty much looked into. But I think it's a potential explanation if we continue to collect the data and see these interesting effects. Yes, they do that in the third session. So in each session is, I don't know if I mentioned this, but each session is separated by at least 2 weeks.
And what do you tell them to do during that?
So we tell them that they're going to be presented with, videos. I don't remember the exact instructions, but, they're told to watch the videos and listen to the words while fixating on the black cross at the center of the screen. So the cross shows up when there's no video present to control eye movements, but we keep it that simple.
Yeah, so they're not doing any.
No, they're not doing
Period setting.
No. I will say that we are, partially because of the clinical nature of our populations, I check in with them every 10 trials on the EEG task, to make sure that they're still paying attention, that they're comfortable, etc. Need to go to the bathroom. And I think that generally does kind of help keep them on task because I often encourage them to stretch their legs, and take a frequent break.
Do you think at some point you'll do some analysis where you're just focusing on the items that are more
Perfectly. I feel like we have very few items that are perfect. Plus, yeah, I defined it based on the difference between the ratings. So it depends on what we define as perfect there, but we could also pick the targets. I see what you're saying. Pick the targets where they got a 7, where they got the highest point on the Layham scale.
or whatever.
Yeah. I could have a look at that. We might end up with I didn't necessarily explore that route because I had maybe erroneously assumed that we would end up with too few items.
Or you can always include that as a covariate.
Yeah. Yes, we are. When we will be using mixed effects models to analyze this data when it's fully collected. And one of the purposes of the norming, in addition to item selection up front, is to include it as a covariate in the analyses. Yes.
I just have a follow-up question to what Erica said. So about the EEG task. So if participants kind of didn't know what they should do, like they should just listen and watch, right? I'm wondering whether it's possible that the priming actually swapped. So for example, the participant with Parkinson's disease, maybe they, forgot what they were doing and they were looking at, first they were hearing the word and then they were looking at the next trial, the video of the next trial. And this is the effect you're actually observing in that participant, which will be incongruent potentially.
Potentially, though it would be, given the way that we randomize the trials, for them to consistently show a difference between related and unrelated trials, I think, if that's what they were doing, I think that would be a little unlikely.
My point is that, maybe, if there was no specific task, if they were instructed that pay attention to the video and then match if the next word is matching or not, then, they may be looking at, oh, does the word match the next video? You know what I mean? So they may just be doing something else than you were expecting.
Yeah.
That's just a thought. I don't know.
No, it's a fair thought.
But usually we do have something like trials, but they're not the experimental trials.
Right. Yeah, we we were going to originally do that. And then we piloted it with a couple of older participants, and we collectively at the time came to the conclusion we thought it was too disruptive and distracting for them, but we did go back and forth. Because yeah, I do also have some concerns, but like I said, I'm generally encouraged by the fact that there does seem to be a differentiation in the related trials. And for most of the individuals presented here, it is in the direction that we would expect.
So you said that the EEG task is before the behavioral task,
Correct.
and I was wondering if it's the same set of objects and same pairings?
It is.
Okay, so do you think there's some type of priming that could be going on from the exposure to the behavioral task where they can prep themselves kind of what's gonna be?
I mean, potentially, they're presented with quite a few items, not consistently in the same order. And each target only has one related, one unrelated pairing. So especially given that we've spaced them out, I think it's, yeah, I think it's unlikely. But there is a potential for that. That's actually why we put the trials first, just because we wanted to make sure we weren't diminishing the possibility of observing that 400 by prior exposure to the stimuli. So yeah, that could potentially have consequences for the behavioral version.
I might have missed this, but how many trials were they performing in each session, and how many were retained with EEG being so noisy?
Yes, so we were presenting 160 trials in each session. So we had 2 sets of 80 trials each. 2 lists, we would take a break, a forced break in the middle, even if they didn't want it. And then we would repeat the same items a week later in session 2, like I said, just kind of double the data that we ended up with. So I will say the first PPA participant that we looked at I had to throw out quite a few trials, which is probably why their signal's so noisy. For most participants, it was less than 15% of trials. I think.
Do we have like a minimum number of trials that need to be retained to obtain like a
So
stable?
the upper end for neurotypicals is 25%, and I'm staying well below that, and that's the aim. For clinical trials or clinical populations based on my recommendations from the EEG training camp I went to and other researchers, 50% is the highest we're willing to go. But other than that one participant, and he wasn't even quite that threshold, the data's been actually surprisingly good. I'm trying not to jinx myself, but I think that with the frequent breaks that we've incorporated and just kind of, the other thing that the breaks lets do is if an electrode starts acting up, we can intervene and adjust it, right? So I think that particular design feature has allowed us to collect surprisingly quickly.
Interpretation. So you're hypothesizing that there's going to be a muted response of the hippo and potentially a delayed response.
Yeah, that based on the prior literature, that's what we would expect. I will say though, that in these populations, particularly in both of these populations, really, there's not a ton of EEG research looking at the specific topic of interest. So there's not a ton of N400 studies in either population of interest. So I didn't have a lot of prior findings to kind of base the ERP predictions. So, but yeah, I mean, logically speaking too as well, we would expect to reduce if we see reduced evidence for behavioral prevention. But so I have the plots, but I want to talk to Brian Erickson before I present them to anybody to make sure I'm not misrepresenting. And I'm also still trying to figure out how to kind of group across the verticals. I hope to have that done for this talk, but